Hi everyone, I'm going to review chapter 11.5, which goes over cancer and cell cycle regulation. And in this lesson, I will spend a little bit of extra time towards the end going over some aspects of cell treatment that students commonly have questions on. But at that point, it'll go beyond what's required for your notes, and I'll signal when that happens so you can kind of skip over that section if you so desire. So, some reminders about cell cycles and how we control and make sure that things are done in the proper way. So, we want to make sure our cell cycle only progresses to the next stage. We only enter G0 into S phase when our cells are in the appropriate stage where it necessitates division. We only enter from S and G2 into the M phase when all our material is ready to go and our DNA has been checked that it's fairly good. And we only finalize mitosis when we double check our DNA and sister chromatids have separated correctly. All these things occur because once you go beyond those points, if there is a failure at the next stage, your cell can no longer get back. It oftentimes may end up dying or arresting, meaning that cell kind of is all scrambled a little bit and it can't really ever continue on in the cell cycle. All right. And all those points where you double check your work before moving on to the next stage, that's the checkpoints that were mentioned earlier in chapter 11.1. So checkpoints ensure cell division occurs with minimal issue. It ensures that you don't progress until things are correct. So they're basically stop and go signals. You stop the cell cycle and you stay in the stage you're in or the phase you're in if you are not ready to move on, and you go if all indicators show that you're ready to divide or that it's appropriate to divide in that moment. You don't want your cells to divide when you don't need new cells, for instance. So, because of that, because you're regulating it, it's reliant on signals that indicate if cellular processes have been completed correctly. A lot of the time, the signals or the ability to respond to the signals are done by proteins, and these proteins are coded by genes. So what is actually happening is those checkpoints we talk about are regulatory genes. And because they're genes, they're DNA, that means that they can be mutated and stop functioning. And that's what we're really going to be talking about today. What regulates the cell cycle? What are the genes regulating the cell cycle, and what happens when they stop working? Because if you mutate those checkpoint genes, those genes will then potentially lose their function, and they may potentially fail to regulate those checkpoints. And if you make too much or too little of a stop or go protein, a protein that helps you stop the phase or go in your phase, then that can lead to major problems. And the main problem we're going to talk about today is cancer. So, generally speaking, if the things are mutated in such a way so that your cells can't continue division, that generally isn't going to be a problem because you have so many cells in your body. If one cell is damaged and unable to continue dividing because it accidentally got a mutation, but the other cells have DNA that's not having an issue, those cells will replace whatever issue occurs with that new cell. The problem really occurs when a cell adopts enough mutations and overrides enough checkpoints that it can start dividing uncontrollably. It can start skipping over or speeding through interphase so that division occurs over and over with little to no resting period. And at that point, if your cells keep dividing, they actually accumulate more and more mutations that make them more and more abnormal, and they start building up and overcrowding other cells around it. And that is basically cancer. So, what is cancer? Well, we want to make sure we understand what is the difference between cancer and a normal growth, abnormal growth, because not all abnormal growths are cancer. Some of them are just considered a benign tumor. So we want to know some terms. So neoplasm is the accumulation of abnormally dividing cells. So oftentimes I use the term neoplasm and tumor interchangeably, one or the other. However, some slight differences is when we say the word tumors, we're saying that the neoplasm has grown enough that there's a visible or noticeable lump in the tissue that's of an abnormal density compared to the surrounding tissue. So a wart is a classic sign of a benign tumor or benign neoplasm, where you can definitely feel that the texture of the wart is different than the surrounding tissues. Now, benign neoplasms or tumors are growths that are not cancerous. So they don't move from the original location. They tend to be slow growing lumps, but they don't take over tissues around it or spread around your body. So most moles are examples of some kinds of benign neoplasms, and warts especially, but there's other kinds of benign neoplasms. The ones where we really worry are malignant neoplasms or malignant tumors. So you probably know the word malignant, or mal means bad. So malignant neoplasms or tumors are the actual things that we consider cancerous. They're cells that can have the ability to be cancerous because not only do they grow, but they can spread and take over other tissues and move through your body and start growing elsewhere in your body. The actual process of cells actually growing in a different spot, we call that metastasis, and that's actually associated with stage 4 of cancer. All right, so malignant neoplasms are growing cells that have the ability to spread, even though they may not have spread throughout your body yet. But the point where they can start detaching and moving and growing elsewhere in different parts of the body versus the area where they originated, that's what we call metastasis. So you can kind of see it. These are different kinds of moles, but the mole on top here is benign, and you can kind of see it's kind of staying in its place. But over here, these moles, which are associated with melanoma, you can kind of see how they're fading into the tissue around it, and that's indication that it's taking over the healthy tissue surrounding that mole, and it's not just staying in its spot. So a malignant neoplasm gets progressively worse and is dangerous to health if you don't stop it and recognize and stop it as soon as it occurs. So characteristics of malignancy is abnormal growth and development, and oftentimes that's also associated with altered cytoplasm and plasma membranes that allow it to take on those crazy characteristics of being able to spread around the body and not just grow where they're located. A lot of your tissues in your body grow where they're supposed to be located. If a cell in your heart suddenly moves to your brain, it's not going to suddenly start growing heart tissue in your brain. All right? If you swallow a nail, you don't suddenly start growing skin nails in your stomach. Your cells grow where they're supposed to grow, but in these cancerous cells, these malignant cells, they don't have that limitation. And as a result, those cells divide uncontrollably. They're not regulated, so mutations keep occurring and they accumulate rapidly, and mutations continue unchecked, and the cancer can progress. And maybe it started a certain way, but then over time it can start becoming more resistant to drugs or spread faster than it may have initially started. So to see if a tumor that's in someone's body is a benign tumor or a malignant tumor, we often do what's called a biopsy. So a biopsy is when you insert a needle into wherever the tumor tissue is, and you remove a little bit of the cells from that suspected tumor, and then you analyze the growth of those cells and the DNA of the cells, and you see if it's cancerous from that point. If those cells show to be benign, meaning they're overgrowing but they're kind of in the same location, then the medical options for treating it won't be as aggressive, and because a lot of the aggressive techniques for treating cancer can be pretty damaging to the body as a whole. When we call something cancer, it's when you test something and you realize it's malignant, and it's just the actual resulting disease that's caused by those malignant neoplasms, aka cancers, because when they're able to grow and take over other tissues, that creates all kinds of health outcomes that are a problem. So we call that cancer at that point. Okay, so what makes something a cancerous cell versus what makes something not a cancerous cell? So all healthy cells have two characteristics, and they have both these characteristics. They have what's called contact inhibition or density-dependent inhibition, which is the fact that cells stop growing once the space they're in is filled. So what that means is these cells, when they finally fill their space, they don't keep growing, they kind of stop until you remove cells from that layer, and at that point, they'll regrow and refill that area. So that's kind of a characteristics controlled by both signals in the cytoplasm and proteins in the cytoplasm and the cell itself. Anchorage dependence also relies a little bit on the cytoplasm and cell itself, because in this case, that's the ability for cells to stay where they're located and not move and grow elsewhere. That's the whole point where I said if suddenly a heart cell got detached and moved to your brain, it doesn't start growing a heart in your brain. That idea. Okay? Sometimes people note it's a substrate. It's substrate dependent, meaning it has to be attached to a substrate to grow. Substrate just refers to molecules that attach to proteins, because you have proteins in your membrane, and when they anchor to the appropriate location in your cell, which has like probably some protein or other molecule it has to attach to, the second cell will not hang out with it unless it has the appropriate molecule it's supposed to cling on to. Okay? So they have these proper membrane, what we call adhesion proteins, those proteins that help them cells glue to other cells properly or other locations properly. So this is a classic example of a lack of contact inhibition. So in this case here, you override the signals that stop your cells from continuing to grow even when you have enough cells to fill the space. Now, that's not necessarily a problem. This is something that happens in any tumor, benign or malignant. It's just that they kind of overgrow. But that's just a tumor. It could be a benign tumor, and those only have that mutation. Again, the problem with cancer is not only is it overgrowing, it's taking over other healthy cells. When you have a benign tumor, it only becomes a problem if the tumor grows large enough that starts pressing on other tissues. And if you like gross surgical videos, you can look at massive tumor removal videos if you'd like. But generally speaking, because they don't spread into other tissues, they're not going to overtake your heart, your stomach, your brain. The problem here comes when you have a cancer cell that not only overgrows because it lacks contact inhibition, but it also has the ability to grow anywhere. It lacks anchorage dependence. It doesn't have to stay in place, attached to the right thing. So at that point, when your cell not only overgrows, but it can grow into any space and any location, that's when cancer cells, spread to your heart, your stomach, your brain, lungs, things like that, and then they interfere with the ability for those vital organs to function. So when we call something cancerous, it must lack both. It must lack contact inhibition, meaning it overgrows, and it must lack anchorage dependence, meaning it can grow anywhere and take over anything. One classic example of a major cancer cell is Henrietta Lacks cervical cancer cells, which are actually a cell line that has been able to continue dividing unlimitedly since it was discovered over 50 years ago. And this cell line has become a really important part of biology research, but there's a lot of ethical problems with how the cells were obtained. So if you're curious about that information, you might actually be into watching this TED Ed video. It's also a little bit more explained in chapter 11.6 of the textbook. It's just really interesting. There's books about it and documentaries about it and stuff like that, especially if you're kind of interested in the space between medical research and ethics. Anyways, and how some groups can sometimes be screwed over. Okay, but what makes cancer cells special? Not special in the good way. It's, again, that ability to not just overgrow, but to spread. So, is this a malignant or a benign neoplasm? We don't know. You have to look at the genes. But if those genes say, or the mitotic tests on it say that it's malignant, that means it has the ability to go through all these stages after here, which means they don't just grow and grow and grow, but they start moving into the tissues underneath, and they can hitch a ride in the bloodstream and find a new place to grow. Okay? So the stages of cancer that you hear about, all of those stages, whether it's small or large, they're still all malignant neoplasms. They all have cells that lack anchorage dependence and contact inhibition. The only difference is how far they've moved. So stage 1 and 2, they've only reached the tissues that are nearby where they started. But once they're finally able to spread and grow large enough to hitch a ride in your blood, that's when you start reaching stage 3 when it initially happens, and stage 4 when it's really gone kind of out of control. So skin cancer is the most common cancer type, by the way. So a lot of students tend to have questions about that. So just by the way, these are different types of skin cancers. You don't need to take notes on this. I just thought it was kind of interesting for those of you who kind of want to know more about different kinds of skin cancer. It's, I think, accounts for 1/3 of newly diagnosed cancers per year. And they say, I think, 1/5 of adults, at least in the US, are likely to be diagnosed with some kind of skin cancer in their life. But most of them are very easily diagnosed and very easily treated, especially these first two. Because these first two have characteristics where they don't spread through your body easily. The melanoma kind is well known because it's, if you have it, it's known for being a really fast grower and the ability to spread very quickly before you can get it treated. Okay, so the genes that affect the cell cycle and those checkpoints, they kind of come under two major categories. There's more categories beyond this, but the two ones we really want to pay attention to are proto-oncogenes and tumor suppressor genes. Okay, so, and we want to be careful we understand the differences between the two. So proto-oncogenes are go signals. They say, hey, cell cycle, divide now, go on, turn on, and continue the cell division process. However, this is when the go genes are, the go ahead and divide genes are working properly. It's turning something on, but it has the ability to switch off. The problem happens when proto-oncogenes gain a mutation, and this mutation makes them get stuck in the on position. So imagine a blender. You only want to blend something for a minute, but now the blender won't stop turning on. It just keeps going and you can't stop it. That is an oncogene. So an oncogene is a mutated proto-oncogene that's stuck in the on position and forces a cell to divide even when it's inappropriate, even when it should stop dividing because of contact inhibition. It should stop dividing because it's not on the proper surface. It's not anchored to the proper surface. But in this case, because the oncogene is stuck in the on position, it keeps dividing even when it's inappropriate. That's what an oncogene is. The normal version of the oncogene, where it's able to turn on, it's able to switch on only when appropriate, that's proto-oncogene. So it's the same version of one gene, just dangerous is the oncogene versus the safe version, which is a proto-oncogene. Okay. The second gene I want to cover is the tumor suppressor gene. So the tumor suppressor gene is your stop signals. They're the stops in the checkpoint that prevent you from moving on when it's inappropriate. So remember, cancer is unlimited division, just constant division. So the cancerous form of a tumor suppressor gene is when it's mutated so it stops working. So proto-oncogenes become cancerous when they mutate so they're stuck on the on position. They're always expressed even when they shouldn't be. Tumor suppressor genes can help cause cancer when they stop doing their job of preventing cell division when it's inappropriate. Okay? So they get mutated so they stop working. So in order to actually have cancer happen in a cell, you actually need both these things to be messed up. You need your proto-oncogenes to become oncogenes, and you need your tumor suppressor gene to be mutated and stop working. When both of those things occur in a cell, that cell is capable of becoming cancerous. So you actually have two ways of preventing cancer from happening, these two classes of genes that really help stop cancer. But when both are messed up, that's when you really get that crazy growth. So proto-oncogenes often, by the way, involve growth factors. Growth factors are the molecules that in those checkpoints that help say, hey, let's keep dividing. And we know that because if you inject some of these growth factors into a cell, you can force division to occur. And then if you inject, likewise, a tumor suppressor thing, you can actually stop cell division. But if you inject growth factors, you can cause cell division. But anyways, that's just kind of messy. But here's a classic example. This is actually cancer tissue. The brown stuff is cancer. The lighter color tissue is not cancer. And in this case here, the cancerous cells are dyed brown with a tracer one, but it's actually showing anything that's overexpressing the growth factor EGF that's coded by oncogene or a proto-oncogene, those it's going to be browner than the other tissues around it. So you'll notice that the cancer cells around here just have way more growth factor expression than the lighter tissue around it. Okay, so if you have cancer, you need two things to happen. You both need an on position. You need your proto-oncogenes to become oncogenes, and you need to stop your tumor suppressor genes. There's a couple famous tumor suppressor genes. You might have heard BRCA1, which is really associated with genetics. If a family has a mutated BRCA gene in their gene line, then you might have a family history of really aggressive cancers in your family, and you can actually get this diagnosed through those gene sequencing companies. They can see if you have a mutation in your BRCA1 gene, especially if you have a family history of cancer. So then it'll let you know, oh, you need to keep an eye on making sure you get screened for cancer each year. Okay? One of the other famous tumor suppressor genes is the P53 gene. So the P53 gene actually stops cell division if your DNA is damaged. So if you get a mutation in this tumor suppressor gene, what'll happen is your cells will divide even if the S phase, the replication of DNA phase, is wildly messed up. And what's going to happen is it actually increases the chances of your cells creating mutations in the cancer and getting more aggressive over time. And it also prevents the cell from stopping cell division and recognizing that, something's gone wrong. And that actual process of your cell stopping something, if something goes wrong, and possibly even destroying itself, we actually call that apoptosis. So there's actually mechanisms that if a cell is truly terrible and can potentially become dangerous, your body is capable of telling that cell to destroy itself, to be destroyed, self-destruct, or self-destruct, so that it doesn't have the potential of becoming cancer. So this is sort of a excerpt on what causes cancer, but it's a little bit long to read about it. But if you're interested, you might want to read it because it's kind of interesting how they describe it. But and the importance of apoptosis. But just to kind of summarize, though, the development of cancer involves the messing up of checkpoints and the accumulation of mutations in those checkpoint genes, right? To get cancer to develop, you have to have all of the following occur. Many things occur. You need your checkpoints to stop working so that you turn off your tumor suppressor genes. Your checkpoints also need to be stuck in the on position by having oncogenes be in the form of the genes that are handling whether or not you continue cell cycle, meaning that you should no longer have your proto-oncogenes. They need to be mutated into oncogenes that always force division. And finally, you need to also lose the ability to destruct and destroy dangerous cells, lose the ability to have apoptosis. When we talk about genes for checkpoints, it's the first two, but there's also genes within those checkpoints that help force apoptosis to occur, meaning destruction of bad cells to occur. Okay, so what causes these genes hits? What causes these mutations to accumulate? Well, you need a lot of mutations to accumulate. It's not just one, it needs several, and it has to land in the correct gene. You get a lot of mutations in your life as you live your life, but a lot of time it affects genes that don't really matter, or even non-coding DNA. But it needs to have several mutations occur at different genes that regulate the cell cycle to get to the point where it becomes cancer. So it takes kind of a lifetime for all those mutations to accumulate. That's why a lot of cancers kind of get diagnosed later in life, after you lived a decent amount of life and been exposed to a decent amount of damage. Carcinogens are materials that increase mutations in your DNA to the point that it increases your chances of cancer. And generally speaking, lifestyle changes can reduce exposure. A lot of the things we talked about before when we talked about mutations, but I also want to point out that anything that increases cell damage increases your chance of having cancer, because a lot of carcinogens increase cell damage, both damage and DNA damage, to increase mutations. But the fact, if you get cell damage constantly to your tissues, you have to replace it. The more often you have to divide your cells to replace bad, broken, damaged tissue, the more likely you're going to eventually develop a mutation that could be a problem. Okay? So not only does UV radiation become a problem because it can not only directly damage your DNA, but also because it's damaging your cells overall in your skin, it increases the likelihood that your skin has to replace itself and potentially, in that replacement, potentially mutate. Okay, so how do we treat cancer? So a lot of times cancer treatments really have to do with catching it as early as possible before it has spread throughout your body. That stage 4 process, that malignancy metastasis process where it's hitched a ride in your blood and hitched a ride to another site, that's when it's really hard to stop the cancer. The earlier you get it when it's still in its site, the quicker you can cut it out and prevent it from growing. Generally speaking, the more dangerous the cancer is, it's not because the cancer itself is dangerous, though that could happen. It's usually because it can't get diagnosed until the later stages. It's almost undetectable in the early stages because it's not commonly checked by your doctor. So what treatments can stop cancer? So remember, cancer is rapid division. So one thing, you want to just surgically remove as much of the tumor as possible because that's got the highest density of crazy dividing cells. So the more you can get rid of, the better. But remember, some of them may have hitched a ride, and any 1 or 2 cells that have hitched a ride become dangerous, so you want to destroy them. And a lot of the treatments we associate with cancer, actually what they're doing is they're taking advantage of the fact that these cells are more likely to divide than normal tissues surrounding it. So what it's actually targeting is cells doing mitosis. So in this case, radiation actually targets cells with radiation beams, and it turns out this radioactive process is more likely to damage cells with chromatids, with tightly dense wrapped DNA, which means they're in the M phase. So even though it's destroying cells, and some cells may be healthy, it's destroying more dividing cancer cells than normal cells surrounding it. Chemotherapy works similarly. So this is just radiotherapy where you get exposed to radiation either directly through like a beam transmitted through you or like right on the site. Okay, chemotherapy works similarly, but in this case, instead of using radiation, you're using drugs. So radiation is kind of nice because you're using a beam, so you're just going directly to the spot where you know the tumor was. So you're damaging cells, but only in the spot you're aiming your beam at. While on the other hand, chemotherapy targets your entire body. Chemotherapy is really important in the event your cells have already reached malignancy and metastasis, where it's already hitching a ride in your blood. Then you want to target all potential cancer cells throughout your body. And in this case, you're going to use toxic drugs that also disrupt mitosis. But again, it's not just disrupting your cancer cells, it's disrupting your healthy cells as well. But the aim here is that you're disrupting more cancer cells than healthy cells so that you're able to better off fight the cancer. And then when you have those recovery periods before chemotherapy treatments, your healthy cells can grow in more. And then hopefully you've caught most of the cancer cells so that the cancer can't grow back. At least ideally what happens in successful chemotherapy. All right, at this point, what I'm going to do is, this is my signal to you that if you wish to stop the notes, stop and move to the end of the video, the last minute of the video that just does a quick sum up, and then you're good to go. This stuff is just special interests that I'm going to go over when it comes to talking about traditional treatments for cancer. You don't have to take notes on this. This is just for those of you who are interested in cancer therapies and want to know more about it. Otherwise, feel free to ignore it and just finalize your notes. Okay, so as I mentioned before, radiation is targeting cancerous cells or cells that are dividing, but it also has the potential to damage healthy cells in that same region if they're mid-division. The difference here is that you have far more cancer cells in the middle of division than healthy cells in the middle of division, okay? And ditto with chemotherapy, but the difference here is not only are you targeting cells in the middle of division, you're targeting them throughout your body using drugs and chemicals, while radiation only targets it in a region that the radiation beam was aimed at. Okay, so this picture is actually a close-up of all the potential proteins that help make up the kinetochores, the spindles, the cytoplasmic region that's involved in mitosis, and the centrosome, and major proteins that are involved mid-replication. So all those proteins are involved in the replication cycle. If you can find a chemical or drug, basically the same thing, but if you can find a drug that disrupts it, that drug can become a chemotherapy drug. All right, so the idea here, and this is like the fantasy land one, the idea here is that you add the chemotherapy drug and all the over-dividing cells die and only the healthy cells remain. But that's actually an oversimplification of the actual idea. The reality is most of the tumor dies, though a decent amount of tumor may be left behind. And again, as I mentioned, because it's targeting healthy cells, or not healthy cells, fast-dividing cells, not only is it targeting cancer cells, but any cells in tissues that tend to divide more frequently are the ones that are most likely to get side effects, also get hit in addition to the cancer cells. So your hair grows pretty rapidly, your skin grows pretty rapidly, the lining in your blood vessels can grow pretty rapidly, and the lining in your stomach and GI tract, meaning your intestines, also grows really rapidly. Since food is moving through it, so it gets damaged, you need to replace those things. And because all those tissues lining your organs and on your skin get divide a lot, those are the things that end up getting damaged the most by chemotherapy. And that's why people lose their hair, they might get mouth sores, they start to feel nausea and vomiting because the lining of their stomach is getting messed up. Constipation and diarrhea because their intestinal lining is getting messed up. They might get some bruising and bleeding because not only are the lining in their blood vessels a little bit weaker, their skin is a little bit thinner as well. So all these things are side effects in the attempt to destroy the tumor. You also end up also having some friendly fire against your healthy cells that are just lying around there, just innocently doing mitosis, not realizing that you're shooting all mitotic cells. So we've kind of gone over the advantages of chemotherapy, but also some of the consequences of it. And nowadays, we actually have, with modern drugs and medication and tools, we actually have the ability to provide much more targeted treatments and much more specific treatments to ensure that we primarily damage the cancer cells rather than other cells that aren't cancerous. And in this case, a big aspect of it is actually relies on the ability to sequence the DNA of your cancer. So that's one of the things I was really surprised by. You can, when they do a biopsy on the cancer, they test the genes in it, and if they spot certain mutations, they can kind of customize the treatments based on the specific genes that are mutated in your cancer. And what that ends up doing is you can pick drugs that are more effective for that specific mutation on your cancer. There's also newer technology that involves immunotherapy, and I'll go into a little bit into that. But basically, you can pick the treatment that we know is more effective against those particular mutations or particular changes in the cancer cells that you or whoever has cancer in particular has. So something else, it's kind of adjacent. It's not quite chemotherapy, it's adjacent to chemotherapy. And it's instead of using just any chemical to attack your cells mid-mitosis, instead what you do is you kind of use techniques to train your immune system to specifically target markers on the cells that are cancerous and attack them as though they were any virus infected or bacteria infestation in your body. So you're using your immune system and training it to attack just the cancer. And in this case, these treatments called immunotherapeutics are really effective, again, as long as you can identify the specific mutation that your cancer has that is unique to just the cancer and not your surrounding cells. And as long as the therapy is available, because there has been a lot of medication that has been made lately, but they're very specific for the cancer mutation types. So a treatment that might work on one person's lung cancer would not work on a different person's particular mutation of lung cancer. Additional issue that comes with these kinds of medications and just treatment of cancer overall is that it kind of can, if you're unlucky, lead to greater risk of or greater pressure for your cancer to mutate. What do I mean by that? Well, I mean, if you leave your cancer untreated, it's going to be bad anyways. But one of the issues here is when you target the cancer, sometimes you don't get rid of all of the cancer. Generally speaking, you only get rid of some of the cancer with each treatment. And for example, chemotherapy is often done over a series of months or even years. But what ends up happening is if you leave enough stem cells behind, these stem cells, or called cancer stem cells, are the special cells that are able to detach and form a tumor wherever they float in the body. Not all the cancer cells in a tumor will automatically form a tumor elsewhere, but these cancer stem cells will. If you don't somehow get rid of all of the cancer with your treatments, what you in fact end up doing is killing the most sensitive cells and leaving the cells that are best able to fight off or ignore or avoid the treatments and leaving those behind. And those are the ones who then replicate and continue on the cancer. And then that can lead to reoccurrence of a cancer, even if you get it all the way down to almost undetectable. Sometimes that lingering cell that stuck around is not just lingering and capable of turning into cancer, but it's also now capable of resisting treatment. That's why sometimes when people say they're under remission, but then the cancer comes back, it comes back in a much more aggressive form that's harder to treat. And that's sort of the origin of drug resistance. It's really evolution. Evolution is where the organisms and the cells that are more capable of surviving are the ones that end up taking over. And unfortunately, for those unlucky people that have cancer, they didn't happen to catch everything. They sometimes end up mutating their cancers so that they become resistant to the therapies they are using. And then later on, they're going to have to switch the drug therapy they're using, or their cancer just keeps progressing and their outcomes are way worse. They're much less likely able to survive. So some people, though, they're fine. And that's sort of why when someone is under remission, meaning their cancer is no longer detectable, doctors continue to monitor them very, very carefully for many years after their cancer is undetectable to just make sure they're spotting for any chance that there might be a lingering tumor causing malignant cell that might start popping up and dividing once more. I have a really good friend who had leukemia when she was a teenager, and it's just something that she is part of her yearly checkup to double check for and screen for cancer that wouldn't be typical of another person's checkup. It wouldn't be as closely monitored as it is for my friend. So, in terms of our cell cycles, it's really important that we regulate it very carefully, and we have lots and lots of genes all involved in the regulation process, because if our cells divide at the inappropriate time, they may mutate and they may divide in such a way that they can basically take over other tissues, which is what we define as cancer. So some of the key checkpoint gene types that we covered were proto-oncogenes and tumor suppressor genes. You want to think of them as on switches for proto-oncogenes and tumor suppressor genes as off switches for the cell cycle, for preventing division. So in this case, what leads to cancer is when your on genes are stuck in the on position. They don't have a way to stop turning on. That is what we call an oncogene. It's always on. It doesn't have the ability to turn off. It's always forcing the cell cycle to divide. In addition to that, you also need the tumor suppressor gene, the off switch, to fail. So in this case, a mutation that stops the tumor suppressor gene or at least some of the major tumor suppressor genes, also have to stop functioning in order for cells to grow unchecked. So you need mutations in both, a stuck on position oncogene and a broken off switch in your broken or mutated tumor suppressor gene. And then when these things happen, that's what leads to the lack of anchorage dependence, which means your cell can grow anywhere, and the lack of contact inhibition, which means your cells will continue to grow even when there's not enough space and it's inappropriate. We take advantage of the fact that cancer cells divide far more than regular cells by in our treatments by using treatments that specifically target cells that are in the active stages of dividing. But then for those treatments, they have some bad side effects. More modern medicine can avoid those side effects, but they're very particular to the cancer and they can't be used in as many cancers. All right, and this was the vocabulary we covered. Bye.